The question comes up, can you mimic these beneficial effects of exercise, intermittent fasting, intellectual challenges pharmacologically with some chemical that will mimic the effect? And the answer is...
Welcome to the Brain Fit Podcast, where we share the cutting-edge science on how you can improve your memory and live a happier, healthier life. My name is Robert Love. I'm a neuroscientist, and I have a medical clinic in Florida where we are reversing Alzheimer's in our patients. Today is the interview with Dr. Mark Mattson. Dr. Mattson is one of the most published neuroscientists in the world. He was a professor at Johns Hopkins Medical School, and he was chief of laboratory neurosciences at the National Institute on Aging. And one of his specialties was Alzheimer's and Alzheimer's prevention and what's going on in the brain. So I dig into that really deep with Dr. Mattson. So here is the interview with Dr. Mark Mattson. I am so excited for our next interview. I'm here with Dr. Mark Mattson. He has a PhD in biology, and he was previously head of the National Institute on Aging in the field of neuroscience. He was the head neuroscience in that department. That's a really, really big deal. And currently he is a professor of neuroscience at Johns Hopkins University, which is one of the top research institutions in the world. And as a researcher, he's had just tremendous impact in the field of aging and neuroscience. He has over 200 citations of his published research, which is a really big deal, with an average of over 206 citations per publication. And just so you know, the average number of publications cited is 0. The next most common is 1, and he's averaging 206 other publications are citing his work. So, Dr. Mattson, it's just such an honor to be here with you, and thank you so much for your contribution to science.
Well, thanks for the invitation, Robert. I look forward to this and trying to educate and hopefully in an entertaining way your audience on what the current thinking is on the best ways to keep your brain healthy and functioning as close to as you can get to the peak level throughout your life.
Absolutely. And so I want you to understand that at the end of this, you're going to have some really actionable steps that you can take to both slow down aging and help prevent Alzheimer's disease. And these are very based in science. And Dr. Mattson's published in some of the most prestigious journals like the New England Journal of Medicine on things that you can do that are specifically going to help slow down aging and even help prevent Alzheimer's disease. So this is going to be really powerful. So first, I'd love to know a little bit about your backstory. How did you get into how did you get into this field?
Well, when I was an undergraduate at Iowa State, I was in pre-veterinarian line. I grew up on a farm in southern Minnesota, and we trained and raced harness horses, trotters and pacers that pull these carts or sulkies behind them. And so I wanted to be a veterinarian. I didn't get into veterinary medicine school when I applied. And so I decided to go on to graduate school, and at first, I was interested in endocrinology, so I did a short master's degree in endocrinology. Then my PhD work at the University of Iowa was in neuroendocrinology, how the brain controls adrenal gland, different endocrine, thyroid, and so forth. And it was during that time I got interested in stress, right? Because your brain is kind of the conduit through which your body responds to stress, at least psychological stress. And then I did a postdoc in Fort Collins, Colorado, at Colorado State University, and I was studying brain development. And at that time, the thinking was that neurotransmitters are chemicals that are released from one neuron and then activate or inhibit another neuron at synapses, these connections between the neurons. But I want to know, do neurotransmitters have roles in the actual wiring of the brain during development? That is, do they play roles before synapses are formed? And what I discovered was that glutamate, which is an amino acid and the most important neurotransmitter in the brain, many of your listeners may have heard of dopamine or serotonin, for example, because of the dopamine's role as a pleasure neurotransmitter and addiction, and serotonin's role in depression and antidepressant effects of antidepressant interventions. But it turns out more than 90% of the neurons in the brain deploy glutamate as their neurotransmitter.
90% of the neurons are using glutamate and potentially other neurotransmitters, but 90% are using glutamate. That's amazing.
Yeah, and the only way other neurotransmitters affect our behavior is by subtly modifying the ongoing activity of these circuits that use the excitatory neurotransmitter glutamate. So anyway, I've written a whole book on this. It's called Sculptor and Destroyer. It comes out next month, and I try to make this intelligible to listeners with an interest. I have a lot of nice anecdotes and historical perspectives. But anyway, so then
for those of you really interested in glutamate, we're going to get into that later in the bonus material, because we can go really deep on that. So please continue, Doctor.
Yeah, and it was at that time I got interested in cell death, and particularly death of neurons, because I discovered, and actually several other scientists at the same time were discovering, that when neurons are exposed to very high levels of glutamate that are not normal levels, they're killed in a process called excitotoxicity. They are excited to death. People know about epileptic seizures, right? That the neurons are firing like crazy, out of control. And what's happening there is glutamate keeps being released from synapses and keeps activating the next neurons. And if that happens too intensely, it can kill neurons. And we think a more subtle, insidious form of excitotoxicity occurs in Alzheimer's and even in Parkinson's and other age-related neurological disorders.
And that's so interesting, because it's counterintuitive. Most people believe Alzheimer's disease is a lack of neurotransmitter activity, and Parkinson's disease is a lack of dopaminergic activity specifically. And then your research and what you're alluding to is that it's perhaps overuse of glutamate or excitotoxicity from glutamate that actually may be contributing to these neurodegenerative diseases.
Yeah. So by the time someone is symptomatic with Alzheimer's or Parkinson's, a lot of neurons have already died. So in Parkinson's, by the time someone has trouble controlling their body movements, and the classic features of Parkinson's, tremor, there are already probably 70 to 80% of the dopamine neurons in that brain region have already died.
Really? By the time someone's symptomatic with Parkinson's, 70 to 80%?
And they're died because they've been activated uncontrollably by glutamate.
If you or someone you love has Alzheimer's or another form of dementia, I want you to know there is hope. Alzheimer's is preventable, and it is even reversible. In fact, we are reversing Alzheimer's in my medical clinic here in Florida. My partner and medical director is Dr. Josh Hellman, a medical doctor trained at Harvard who specializes in preventing and reversing Alzheimer's disease, and we are reversing Alzheimer's in our patients. If you'd like to learn more about working with us to both prevent Alzheimer's and even reverse Alzheimer's and reverse memory loss associated with other forms of dementia, we would love to hear from you and send you some free information so you can learn what we are doing and see if it's a fit for you to come down and work with us in our clinic. We also work with other clinics around the country to help patients both prevent Alzheimer's as well as to reverse Alzheimer's and reverse memory loss associated with dementia. So if you'd like to learn more, go to brainpowerclinics.com. Go to brainpowerclinics.com and then enter your information. We will send you some free information, and then we hope to work with you, if we can be helpful to you, to help you prevent and reverse Alzheimer's disease. Now, is that just in the substantia nigra, or is that dopaminergic neurons throughout the brain?
Well, no, that's a good question. It turns out, that's another reason glutamate's the most important neurotransmitter. In all your brain regions, the core circuitry is glutamatergic neurons that use glutamate, and GABAergic neurons that use the inhibitory transmitter GABA. Neurons that use dopamine are located, their cell bodies are located in two small discrete regions. One's the substantia nigra. It's in the midbrain, a primitive region of the brain. And the other is in a region called the VTA. But anyway, the VTA is associated a lot with addiction, whereas the substantia nigra is associated with controlling body movements in Parkinson's. But it's the same with serotonin. The actual neurons themselves, they use serotonin as a neurotransmitter. They're not all over the brain. They're only in a small region in the brainstem called the raphe nucleus. And they have the axons that reach way up to a lot of neurons up here and modify their function.
So interesting. And so the loss of function in our brain, and this is really important for those of you who want to understand neurodegenerative diseases, Alzheimer's disease, Parkinson's disease, and so forth, is a lot of the loss of brain activity is often due to excitotoxicity, overexcitation due to excess glutamate, and that overexcitation kills neurons, and then there's that deficit of activity. Is that right, Dr. Mattson?
Yeah, and it's probably the main reason that aging is the main risk factor for Alzheimer's and Parkinson's, because during aging, there's changes that occur in neurons that make them more vulnerable to this excitotoxicity. For example, the function of their mitochondria, the energy factory in the cell, declines. And when that happens, this is all my book, so I don't want to go into detail, but the bottom line is when there's like this insidious energy failure that's occurring, and the neurons continue to be excited when they're not getting as much energy as they should, they can be killed more easily by glutamate. And this will come up when we talk about intermittent fasting, actually, because it turns out that the ketone bodies that are produced during fasting, they are a really efficient energy source for neurons. And we think that as we age, the neurons are still able to use ketones pretty well as an energy source, whereas if they start failing, their ability to use glucose is impaired.
Terrific. So let's get right into that. And so before we do that, I'd really like to can I ask a clarifying question? So as we age, our brain cells become more vulnerable to excitotoxicity, and in particular, our mitochondria become more vulnerable. Is that correct?
That's right.
And therefore, we're more vulnerable to losing our energy centers of our cells and just the cell in general. And that is one of the primary risk factors why aging greatly increases the risk of neurodegenerative diseases.
Yes.
So if we can protect our brain cells and if we can protect our mitochondria, that could be very protective and preventative for neurodegenerative diseases.
Absolutely.
Fantastic. So let's get right into it. You've done a ton of research on intermittent fasting and how that's neuroprotective. Can you talk about the benefits of intermittent fasting for the brain and why that is so beneficial for the brain and beneficial for reducing the risk of Alzheimer's disease?
Okay, so I'll try to keep this simple. So if people eat breakfast, lunch, and dinner and a snack, every time they eat, the energy stores in the liver, which is glucose, are replenished. If they're spacing their meals out like that, and they're not exercising, which is an important point too, then their cells in their body and brain use mainly glucose as their energy source. When a human fast, doesn't take in any calories for, say, 12 to 14 hours, which seems to be when this, what we call a metabolic switch, occurs.
Now, does that differ between men and women? Because men generally do better with longer fasts, women do better with shorter fasts. That number 12 to 14, is it 10 for women, or does it vary between the sexes?
Robert, I don't know. I haven't seen any data on that.
Okay, please continue.
But it varies. It definitely varies depending on whether you're getting your level of exercise. So,
okay,
for example, if you get up in the morning and go on a run for an hour before you eat anything, say you've only been fasting for 10 hours by the time you get up and get out on the road on your run, sometime during your run, you're going to deplete your liver glucose stores and you'll get this metabolic switch occurring. If you don't eat breakfast and don't run, it won't be until late morning.
So exercising when fasting increases our metabolic need for ketones.
Yeah.
And then therefore that increases the availability of ketones in the body and then for energy for the brain. I would like to thank our sponsor, Optimizers. They make great supplements that are good for your brain and good for your overall body as well. My favorite supplement that they make is Mattson. This contains digestive enzymes that help break down fat, protein, and carbohydrates. I love taking this in the morning with my bulletproof coffee, which has a lot of fat in it, so it helps break down the fat. And I like to take this at night before my largest meal, which is dinner, to help break down the fat and protein and carbs from my biggest meal. If you'd like to try this, you can try this risk-free and get 10% off. So to get that, go to buyoptimizers.com/love, and you can try this or any of their great supplements and save 10%. So it sounds like that's one of the best things someone can do if they want maximum benefit of fasting, is to do mild to moderate exercise when fasting to boost ketone production and get a bunch of energy to the brain.
There's experimental evidence for that in animals. There's some things coming out in humans. This is a really very active area now from the standpoint of human endurance research. And we can talk about that too. But if you look at this from an evolutionary perspective, which I often do, it's only been very recently in our evolution that we've been eating breakfast, lunch, and dinner, right? We didn't have the luxury of waking up in the morning and there'd be food in the refrigerator or the pantry. And so our human ancestors, hunter-gatherer type situation, and animals in the wild in general, they have to, particularly predators, they have to expend a lot of the energy in a food-deprived state in order to be successful in getting food, right? So it makes sense that individuals whose bodies and brains function well in a food-deprived state had an evolutionary advantage and passed their genes on to subsequent generations. And it turns out a lot of those genes are genes that function in what we call adaptive stress or hormesis-based mechanisms.
So if I understand you, our ancestors fasted by necessity. They didn't have food readily available. Therefore, our ancestors who could succeed in the environment when fasting were likely to pass on their genes. And those genes are also do really well in adaptive stress, like being in a fasted environment.
Yeah, the individuals who were successful had genes that helped them function well in that food-deprived state.
And that's most of our ancestors. Therefore, most of us probably have some DNA that's or some genes that do really well when we're in a fasted state.
Yeah.
So this is programmed into us for fasting, whether it's intermittent fasting or several multi-day fasts. 1, 2, 3 day fast. That's just part of who we are, and that, generally speaking, is good for us because it's just been handed down to us from our hunter-gatherer ancestors.
Yeah, and so I often make the analogy. I often pretend I'm a cell, a muscle cell, a nerve cell, a heart cell, and because this is a really useful way of thinking of these things. So in the case of exercise, pretty much anybody recognizes this, but it turns out there's a huge science underlying this. During exercise, it's a stress on your muscle cells. When you're running fast, it's a lot of stress on your muscle cells, or, pumping iron and you're getting fatigued. And by stress, what I mean is when your muscle cells fire to contract, there's sodium influx, calcium influx, a big increase in mitochondrial activity to generate the ATP, which is required for the actual physical contraction of the muscle. But it turns out these early events, the influx in particular of calcium from the outside of the cell into the cell, sets in motion a number of downstream events, including turning on genes that help the muscle cells resist more stress and be more resilient in future exposures to stress. So it takes time to get in shape with physical exercise, several weeks to a month before you'll be, really clear, obvious effects. And during that time, there's these adaptations that are occurring in response to these cycles of the challenge the stress during the exercise, and then during the recovery period, resting, eating, sleeping, there are things that go on in the muscle cells that they actually get bigger. So your muscle cells don't get bigger when you're exercising. They actually get physically bigger, more mitochondria when you're resting. But if you don't exercise, these events that set in motion what happens during the growth period don't occur. So this is a very important concept, these transient exposures to intermittent challenges or good stresses, evolutionarily normal.
So hormesis, putting ourselves into good stress, whether it's exercise, whether it's fasting, and it might be, dealing with challenges. People in business, or certainly parents, understand that children have challenges they need to solve with their brains, and that's a good activity. And then parents have challenges with children. It's a healthy exercise of our parenting muscles. I'm not a parent. I'm an uncle, and I love spending time with my nephews. It's really interesting to see how they behave and what I can do to influence their behavior in a healthy way. It's really good to see the stress of children and then try to deal with it as an adult, to not shy away from these challenges of the day, but to use them as an exercise of our brain.
Yeah, and of course, not overdo. There's this zone, there's a sweet spot, or medic sweet spot. Chronic psychosocial stress, or working, 16 hours a day, every day, that's going to eventually cause some problems.
Absolutely.
The recovery period is very important.
I think some of your colleagues, certainly neuroscientists in grad school, could use that little lesson to not overstress themselves in the pursuit of science.
And to go, this works for almost everybody I know who exercises, right? Myself, I do a lot of my best thinking is when I'm going on a run or biking, or in the few hours following the exercise. It's invariably I'm cognitively very sharp and kind of thinking in a out of the box or more laid back way, relaxed environment. So I think that that also makes sense from an evolutionary standpoint.
Absolutely. People ask, what's the number one drug and the number one supplement for brain health or preventing Alzheimer's disease? I say, look, I got a really clear answer for you. It's exercise. All the data show exercise is the very best thing for your brain. It's the very best thing for your overall health done correctly. And if we could bottle exercise into a pill, it would be the best selling pill in history, because exercise has so many benefits, from getting healthy blood, or getting blood pumping to the brain, improving cardiovascular health, which improves brain health, improving our metabolic health, which is really important to brain health, and then the healthy stress, the hormesis effect that happens with it, as well as the self-confidence boost, lowering blood pressure, all kinds of benefits.
Mood is a big one. Antidepressant effect of exercise.
Yes, I think it was research out of Australia found that exercise was more powerful than any pharmaceutical drug regarding depression. And so really, if a medical doctor is prescribing drugs for someone with depression, and some of those drugs do work, really the first prescription is exercise and sunshine, right? If you're depressed, exercise, sunshine, then come back.
I experienced this myself about 4 years ago now. Actually, it was just a few months after I retired from the NIH. I used to do a lot of trail running. Then I started to have some kind of moderate knee issues, so I switched to mountain biking. And I had a mountain bike accident, and it was actually not, it was a bad situation. So on my right side, I completely tore my rectus abdominis off my pubic bone.
Oh my goodness.
And I had damage to the adductor, the muscle that holds your knee in. And I had a little bit of damage on the left. And then I ended up having to have 3 surgeries. But anyway, the bottom line for this conversation, the main point is that I had to go several months with pretty much no aerobic exercise. And I got depressed. I mean, it was probably a combination of no exercise plus, I can't exercise, I'm in pain, that psychological aspect. But then when I finally got back to, I'm doing like 30 minutes pretty hard on the stationary bike now. In the late morning, after having not eaten anything, before then, and walking, going up and down stairs, and some other things. But yeah, the antidepressant effect of exercise is amazing.
If you suffer from brain fog, or if you'd like to increase your mental clarity, I have a great supplement called NeuroPlus Brain and Focus Formula that can be really helpful to you. This is a combination of 7 powerful ingredients that are shown to improve memory, improve focus, improve attention, reduce brain fog, and increase mental clarity. I really like to take this in the afternoon if I'm feeling afternoon brain fog, or if I'm feeling tired. This helps give my brain more energy so I can continue to work during the day. This works really well in the morning with morning coffee and lion's mane. This also works in the afternoon alone or with lion's mane. Do not take this at night. It is very stimulating. It's very powerful. This can help reduce or even end brain fog in as little as 3 days. The average is about 2 weeks, and it comes with a 6-month unconditional money-back guarantee, so you can try it risk-free. So if you want to try this risk-free, go to robertlove.net and then click the link that says Neuro Plus Brain and Focus Formula. So you were able to bounce back from the depression you experienced after your injury when you started returning to exercise.
That's right.
That's terrific. And this is something I really want to stress to everyone, including myself, because I could use more regular exercise in my life. I often let things in my schedule sometimes interrupt that. But exercise is just the very best thing for your brain. And the truth about Alzheimer's disease and many neurodegenerative diseases is that they are preventable, and you can reduce the risk by doing healthy things like exercise, like sleep, and then Dr. Mattson's work, intermittent fasting, so many things, so many behaviors you can do without drugs, even without supplements, which can be very helpful. But these behaviors are some of the most powerful things we can do for our brain health and reducing the risk of Alzheimer's disease. Dr. Mattson, can you tell us a little bit more about intermittent fasting, about why this is so beneficial for cognition and for helping prevent Alzheimer's disease?
Yeah, sure. So I mentioned the ketones. So what are ketones? When your glucose stores in your liver are depleted, then what are called fatty acids are released from fat cells in your body into the blood. And these fatty acids, essentially, they're similar to what people may know it from as a medium chain triglycerides that some people take for supplements. So
I love MCT oil. That is one of my favorite supplements for my brain.
So there's these like 8 to 10 carbons with hydrogens, right? Hydrocarbons. But anyway, those fatty acids are the immediate precursors of ketones. So these fatty acids go into the liver and they're converted into 2 ketones. One's called BHB, or beta hydroxybutyrate, and the other is called acetoacetate. And we've done a lot of work, and actually most of the work in terms of the role of the ketones in beneficial effects of fasting on the brain come from work with BHB. So BHB goes in the neuron, it's actively moved into the brain. There's this mechanism that moves the ketone into the brain, into the neurons. In the neurons, it goes into the mitochondria, BHB, the ketone goes into the mitochondria, and it's used to produce ATP. It's more efficient than glucose, and less free radicals are produced.
That's really interesting. So glucose, if my understanding, one glucose molecule can produce under aerobic respiration approximately 36 units of ATP. Can you tell us the efficiency of a ketone, and then how that relates to the waste products produced by making?
One molecule of the ketone BHB will produce, I don't know the exact number. It's like 50 some ATP molecules. So it's more.
Yeah,
significantly more than glucose,
and less waste products, therefore less stress on the mitochondria by making energy.
Yeah, and then though it's another very interesting thing is these ketones. They not only an energy source for neurons, but they have a signaling function. They affect gene expression. They activate certain proteins that are called transcription factors that turn on genes. And we did some work on this. I had multiple people in my lab work on this, and they showed that BHB stimulates the production of a protein called BDNF, brain-derived neurotrophic factor. BDNF is a protein that plays critical roles in learning and memory. It's important in the formation of new synapses in the brain. It's important in this process of neurogenesis, where new neurons are produced from stem cells, at least in a couple brain regions, the hippocampus being one, which is a brain region that's very important for learning and memory. It's a brain region that's ravaged in Alzheimer's disease. So the ketones are energy sources, and they turn on genes that do good things for neurons.
So increasing BDNF is a really, really big deal. By the way, BDNF, it's also increased from exercise as well as lion's mane, which is a medicinal mushroom that's been shown to be helpful for cognition. So this is really
interesting. Before I had this, a few years before I had the mountain bike accident, and actually when I was mountain biking, I'd forage for mushrooms.
Oh, really?
Yeah. So I did a lot of foraging for mushrooms like oyster shell and chanterelles, and yeah, so that's a lot of fun. And you're right, those are healthy things to eat.
Have you seen the research out of Singapore? I think it's by Li Fang and colleagues about how 2+ servings of mushrooms per week reduces the risk of Alzheimer's by approximately 60%.
No, and if it's human data, I don't believe it, because how do you do that study? You have to do it over decades and decades and decades.
It was a cross-sectional study where they it's an epidemiological study. It was not a randomized controlled trial, but it just showed that those who eat mushrooms had healthier brains at the end of the study. There's
an association between eating those mushrooms and having a healthier brain, but that doesn't establish cause and effect.
Yes, I agree with that. The data was pretty compelling, though. It's very interesting, especially because we know so little about mushrooms. We just know that they're good for us, right? And it's just like
no, and it's not just. I do have kind of a pet peeve a little bit with epidemiological studies, because they often lead to spurious things coming out in the news that, X is associated with Y, and then when it comes out in print, it's X causes Y, or X reduced the risk for Y.
I agree with that, especially around food. All the epidemiological data about low cholesterol, low fat diets it equals better health, and that's just generally not true. We now think it's like the high carb diets that are really dangerous for the brain. High carb is associated with high triglyceride levels, and then dietary cholesterol really doesn't seem to be associated with plasma cholesterol long term. And so I agree with you about those epidemiological data from the 80s and 90s that a lot of conclusions were drawn from. Are not super helpful. So if I understand intermittent fasting, the benefits of intermittent fasting, number one, it's in alignment with our evolutionary behavior. Our ancestors did well with intermittent fasting, and therefore we do well with it. Number two, ketones. Ketones are more energy efficient in the brain, and they produce less waste products than using glucose for energy in the brain. And then you said this in another podcast, the brain older brains are less able to take up glucose, especially with levels of amyloid plaque in the brain. But that's not true with ketones. So if someone has amyloid plaque in the brain, they're less able to use glucose for energy, but they can use ketones for energy just fine. So it can get energy into the brains of those with plaque in the brain, which is really, really great. And the benefit of ketones affecting gene expression. So BHB can increase the production or the activity of BDNF, brain-derived neurotrophic factor, and that's great for improving just brain function. And it's really important in memory. Is that kind of sum up just
yes.
Some
and
of that stuff?
Mood.
Oh, tell us about mood and intermittent fasting.
Well, so we did some studies. I'll kind of give your listeners a little background in this. So we started studying intermittent fasting in the 1990s, and asking a simple question. At that time, there were some studies showing that if intermittent fasting can extend lifespan in rats and mice. And we knew that age is the major risk factor for Alzheimer's, Parkinson's, stroke. We had some animal models that were relevant to those disorders. And we found that if we put the animals on intermittent fasting, it protected hippocampal neurons against dysfunction and degeneration in the Alzheimer's model. It protected dopaminergic neurons in the Parkinson's model, and it reduced the amount of brain damage and improved functional outcome in stroke. And then what happened, we we did a lot of work looking at the mechanisms, what's happening, why is intermittent fasting having these neuroprotective effects? And one thing we found is that it protects neurons against excitotoxicity by enhancing the activity of GABA using inhibitory neurons. So in other words, animals on intermittent fasting, neural circuits in the brain are less prone to hyperexcitability and excitotoxicity. The neural circuits are better able to constrain activity within normal limits.
So intermittent fasting is neuroprotective.
Yeah.
That's fascinating. In my experience of being in ketosis and doing intermittent fasting, I'm much less likely to get like angry or triggered. But when I eat a bunch of sugar, which I do rarely now, I'm much more likely to fly off the handle. I'm much more likely to become impatient or just feel this tremendous emotional upset roaring in my body. But if I'm fasting, or if doing a healthy fast, if I'm not like super duper hungry, or if I'm doing in ketosis, I find my mood is just much more stable.
Yeah, that makes sense. The first drugs that were developed to treat anxiety were drugs that enhance GABA signaling, Valium. Valium activates GABA receptors, which inhibits neurons. So anyway, the point is,
glutamate and GABA are like two sides of the same coin.
Yeah.
Right?
Yeah.
And so listener, I want you to get glutamate and GABA kind of different sides of the same coin. And so glutamate is excitatory, GABA is inhibitory.
Yeah, that's right. That's right. So, and we noticed this in our animals too, that we have these ways to evaluate the anxiety level of rats or mice. They're very simple things, evolutionarily explained, I guess. For example, we have essentially a box, like, I don't know, 2 feet by 2 feet box. And then if you put a mouse or rat in there, they tend to stay around the edges next to the sides of the box. They don't go out in the middle. Okay, they don't want the hot. They've evolved to naturally stay away from open areas because that's where a hawk can swoop down.
So the middle could be dangerous, and so they're staying out of the middle.
Now, of course, I have an explanation for an intermittent fasting. Okay, but it does make sense. So anyway, animals become less anxious when they're adapted to intermittent fasting. They spend more time in the open area. But this kind of makes sense because if you haven't gotten food for a long time, you may have to take some risks.
More exploratory. So I wonder if intermittent fasting increases cognitive flexibility, right? If they're more able to hold interesting ideas, or if they're more curious, if they come up with more answers to questions. So, a question, what name items that someone would keep in a purse or backpack? Does someone intermittent fasting, do they come up with more answers, more novel answers, for example?
Those are fantastic questions, Robert. And one area that's lacking from a lot of research on intermittent fasting is the brain is human studies, right? So these are the kinds of studies, should be done in humans, exactly what you said. Your psychology friends who are still doing experiments, connect with them, because these are, we actually did a study at NIA, and it's done now. The results are being written up. I can't talk about it, but we took people between the ages of 55 and 70 had obesity and insulin resistance. So aging in general is not so good for your brain, but we can't avoid that too much. Obesity and insulin resistance we can largely avoid, and very bad for the brain as you age. So we randomly assigned those subjects to either 5-2 intermittent fasting, 2 days a week, eating only like 500 calories each day. The other 5 days eat normally. And then, so the metabolic switch will go for 2 days, essentially, a week. And then the control group, we just gave them advice for healthy eating. At baseline, before we started them, on intermittent fasting or not, we did a battery of cognitive tests working memory, word recall, all these kind of standard cognitive tests, anxiety and depression scales. And we did both functional and structural MRI.
Oh, wow.
Yeah, so we have data on neural network activity in their brains and sizes of, although we didn't really expect to necessarily over 2 months. It's only a 2-month study to see.
That's a lot of data you have on some humans. That's great.
Yeah, and we've got blood and cerebrospinal fluid, and we're measuring BDNF, a lot of other things in the cerebrospinal fluid. That's the fluid that bathes the brain.
Is that the best way to measure BDNF? Can you measure it in the blood, or can you measure it
Well, it turns out it's interesting. Neurons aren't the only cells that produce BDNF. For example, heart cells, cardiac cells produce BDNF. So the data on BDNF in the blood is a little mixed. There are some studies reporting exercise increases BDNF in the blood. That may be so, but we can't be sure, is that coming from the heart or the brain or...
Is BDNF, will that cross the blood-brain barrier?
Not very well. It doesn't. So it's probably mostly coming from the heart and maybe some other peripheral sources in the blood.
So the best way to measure BDNF right now is cerebral spinal fluid.
Yeah, that's readout of brain levels of BDNF.
Dr. Mattson, I'd love to understand from you acetylcholine. So acetylcholine is a neurotransmitter. I like to call it the master molecule of memory. It's one of those important neurotransmitters involved in the hippocampus, the memory center of the brain. And then, in our society today, so many people want a quick fix. Give me the pill to increase dopamine. Give me the pill to increase memory. Give me the pill to increase acetylcholine. I was wondering if you could talk about some healthy behaviors that people could engage in to naturally increase acetylcholine activity in the brain.
Aside from taking choline, as you mentioned before, mental exercise, mental gymnastics, playing chess, doing what we're doing now, learning new things, not just sitting in front of a TV watching something and not.
So hopefully all of y'all at home have notepads. Hopefully you're learning right now and engaging with this material.
See, and the reason is it's exactly as you indicated before. When you're learning things, there's more acetylcholine being released onto the glutamatergic neurons in the hippocampus. The actual memories are encoded in the glutamatergic synapses, the synapses between the glutamate neurons. But the strengthening of those synapses to an optimal level requires acetylcholine also being released. Okay? So exercise, it doesn't necessarily have a great effect on acetylcholine as, say, it does on BDNF and glutamatergic neural activity. Intermittent fasting, nobody's looked. Well, we looked indirectly, but it was like you'll be really interested in this. Okay, heart rate. Your heart rate beats faster when your sympathetic nervous system is activated, like it is when you're exercising a lot or when you're under mental stress. Your sympathetic nervous system is activated, your heart rate goes up, blood pressure goes up, you actually sweat more. And then heart rate is slowed by the parasympathetic nervous system, which through the vagus nerve, those axons release acetylcholine onto the heart muscle cells, and that slows heart rate.
Now, the vagus nerve also connects the gut and the brain, correct?
Absolutely. Yeah, the vagus, I should mention my podcast here. I have a podcast called Brain Ponderings. I have a YouTube channel. It's on Spotify and other audio outlets. And I had a podcast on the vagus nerve and all these studies looking at stimulation of the vagus nerve. But there's this bidirectional communication through the vagus nerve between your peripheral organs, say your gut and your brain. But to get back to your point on how to boost acetylcholine, we think intermittent fasting might. We did these studies where we put electrodes in the heart and aorta of, actually we did EEG too in the brain of rats. And then we recorded their heart rate, blood pressure, and EEG activity. And then we put them on every other day fasting regimen and saw what happened. We could monitor it 24/7. So we're looking at heart rate, blood pressure 24/7 over periods of weeks and weeks and weeks. And what we found is within 2 weeks of the initiation of intermittent fasting, there was a reduction in resting heart rate, reduction in resting blood pressure. And then we did some kind of sophisticated analyses that showed what's happening is there's increased activity of the acetylcholine neurons in the vagus nerve that slow heart rate. And this is exactly the same change that occurs in endurance athletes. They have slower resting heart rate, blood pressure, but their hearts are more adaptable to stress and more able to resist stress. And in fact, we did a model of myocardial infarction and found that the rats on intermittent fasting had less heart damage and better function of the heart after we give this. We just do occlude the coronary artery, right? We just essentially tie off blood flow to the heart coronary artery for an hour.
I would like to thank our sponsor, Microingredients. Microingredients has really high quality products. All of their products are third-party tested, which means an independent company has tested them for quality and safety. So you know you are getting the highest quality products. They have a great collagen. This great anti-aging benefit helps reduce skin wrinkles, great for your skin, hair, and nails. MCT oil is terrific in coffee, great dairy replacement. And this is also great for the brain. Can even help with weight loss. Their blueberry powder, their organic blueberry powder. I'd love to add this to my smoothies. And their newest supplement, their curcumin supplement, is fantastic. So a good curcumin supplement will have 1,000 milligrams a day of curcumin, and it will have black pepper. So this has black pepper, and this has 1,000 milligrams of curcumin, plus some other things in it, and MCT oil as well, which helps with absorption. Research shows, I've been taking curcumin for at least 5 years. I'm going to take curcumin as long as I'm alive until the science changes, because this is so good for your brain. Curcumin helps reduce the accumulation of amyloid plaque in the brain. Curcumin improves memory in double-blind, placebo-controlled studies. Curcumin reduces inflammation. It slows down aging. Is one of the safest, most powerful supplements you can take. And so if you want to get this from Microingredients, go to Amazon and then use the coupon code RobertLove at checkout, and that will get you a really nice discount on curcumin.
Now, myocardial infarction, that's basically a heart attack.
Yeah, heart attack.
So mice given heart attacks, those doing intermittent fasting, they had less damage to their brain or less damage to their body?
No, to their heart.
So less damage to their heart.
Yeah.
Then I just want to make sure I understand the earlier research. Intermittent fasting increased acetylcholine activity in the vagus nerve?
That's right.
Okay. So intermittent fasting may increase acetylcholine activity in the brain.
That's my point. Yeah.
That's really interesting, because then it'd be tied to learning and memory, potentially learning and memory and other activities of acetylcholine. I wonder if it affects motor movement. There's, I think I read an article that sprinters will take acetylcholine, like choline precursors, because acetylcholine is involved in motor movement. And so by ensuring they have enough, choline, that ensures that their synapses are firing correctly. So this is my gesture of motor movement. Look at my fingers. This great motor movement, right? So having enough choline.
Yeah, acetylcholine's the neurotransmitter released from your motor neurons onto your muscle cells. So that kind of makes sense.
Very interesting. And so I wanted to ask you about GABA. And so you mentioned that intermittent fasting protects against excitotoxicity. Excitotoxicity is basically too much glutamate, and that can harm neurons, make them more vulnerable to cell death and eventually lead to cell death. GABA protects against that, and intermittent fasting increases GABA activity. Is that correct?
That's right.
Now, can someone, and I don't want to encourage not doing intermittent fasting or not exercising. I'm guessing exercising also increases GABA because it reduces stress, and it probably impacts that as well. Another one of the amazing benefits of exercise. Would taking a GABA supplement, would that also have a neuroprotective effect? And then I'd love to know your thoughts. I've seen research, GABA crosses the blood-brain barrier. GABA doesn't cross the blood-brain barrier. The research is kind of mixed on this. I'd love your thoughts on, would a GABA supplement have neuroprotective effects? And what are your thoughts on GABA's ability to cross the blood-brain barrier?
It could have neuroprotective effects. The thing I'm worried about is it may run into the same side effects that people have with taking Valium or, that the drugs. When you take something that way, you're swamping. You've got all these GABA receptors on every glutamate neuron, every excitatory neuron has GABA receptors, the proteins that mediate the inhibitory effects of the GABAergic neurons. So if you swamp all of your excitatory neurons with GABA, that's not a good thing, because now the brain can't properly control the activity. You're going to have problems with learning and memory and other things. And that's what happens in people who take drugs like Valium and other GABA receptor drugs. I think this is another pet peeve of mine. What has to do with drug companies. So,
don't go pick on the kind drug companies that are just out for our well-being.
Okay, so here's an example. And so there's a drug. Valium is its actual name's called diazepam, but the company marketing name was Valium. Okay, that was the first anti-anxiety drug developed back. It must have been in the 50s, or I can't remember, 50s or something. I can't remember because I wasn't born until 1957. But anyway, most patents for drugs last for 20 years. Okay, so what companies do when a drug goes off patent, what that means is that generic drug companies can produce large amounts of the same drug and sell it at a much lower price. So then what companies do is they make a drug that's essentially does the same thing as the initial drug, oftentimes by just making one little change in the same molecule. And then they do a clinical trial. And of course, it has a benefit because it's doing the same thing, or it has an effect, clinical effect. Of course it does, because it's doing the same thing the previous drug had. And then they have these marketing, they bombard the markets with and claim this is better somehow. And then they sell it for a lot higher price. And they have people go to the doctor's office and kind of schmooze with them and so on. So this has happened with so many different drugs over the years.
I would say, and I want to take this time to acknowledge you, Dr. Mattson. I so appreciate that you are willing to take a professional stance against the current, I'll say, situation or model in the United States, where this is the richest country on planet Earth, and we have the most expensive healthcare system, and we are some of the sickest, least healthy people in the industrialized Western world, and we're paying more money than anybody else. And we have a decreasing lifespan, at least we have for the last 5 or 6 years. And I so appreciate you speaking truth to power and saying, look, I think it's a problem with the food that we're eating and the big food companies, the monocropping companies. I think it's a problem with the medical industrial complex pushing drugs on us that are expensive that we might not need, and talking about the healthy natural solutions like fasting and like exercise. And I so appreciate you going out on a limb and speaking the truth about these matters so that people can learn about them, and that way they can live healthier lives.
Yeah, an ounce of prevention, right, is worth a pound of cure. I did a lot of work on Alzheimer's disease from the very beginning of the amyloid story. My lab showed that this amyloid, when it's accumulating on the surface of neurons, can make them vulnerable to excitotoxicity. And this was way back in the early 90s. And we even did some collaborations with a drug company with some amyloid antibodies. And some of them seem to kind of protect the neurons a little bit against the amyloid. Other antibodies made it worse. And at that point, I was already kind of not too keen on this idea of giving patients antibodies against the amyloid protein. The reason I mention this, very recently, the FDA has approved 2 drugs for Alzheimer's disease.
Yeah, let's talk about those. Aducanumab, and then the other one is Leqembi.
Yeah, these are the company's names. They're both, what they are is antibodies that selectively bind to amyloid. That's what they are. They're called monoclonal antibodies, and there's a whole science on how you make this. But anyway, your immune system produces antibodies, right? When you get infections, right? And that helps the immune system recognize the infectious agent, virus, spike protein, whatever, and clear it out or stop it from proliferating. And so essentially what these antibodies are doing is causing brain inflammation. In the clinical trials, they're saying they get like slowing of the decline of Alzheimer's disease by 30%. But then there's a number of patients that had very severe, bad inflammation of the brain.
And basically brain bleeding, right? I've heard from Dr. Heather Sampson. And you got to monitor them, make sure they don't get brain bleeds.
And one of them died, and moreover, the long-term effects, the clinical trials were done over, I think it was over a period of a year. I mean, that's a reasonable time period, but you got to remember that the course of Alzheimer's disease for average person that gets Alzheimer's from the time they're diagnosed until they die is about 10 years, progressive decline. And so personally, if I was diagnosed with Alzheimer's today, I would not take the antibody, get the antibody treatment. But these companies have invested over decades, literally billions of dollars in developing, this approach based on giving antibodies. And
the amyloid plaque hypothesis that, amyloid plaque was found in Alzheimer's patient 0, Augusta Dieter, and therefore that's what Alzheimer's is about. And so therefore, if we want to prevent Alzheimer's or stop it, we got to reverse the plaque. And that just hasn't turned out true in the data. And yet we still got these giant companies with tons of money who are doing massive amounts of research still on this hypothesis that it's clearly a flawed hypothesis, because there are people with memory loss and Alzheimer's symptoms without a bunch of plaque. There's people with a lot of plaque who don't show cognitive decline, and yet they're still and then Dr. Dale Bredesen points this out in his great research. Amyloid plaque is protective. Amyloid plaque is your brain's natural response
in a way
to inflammation. To certain things will increase plaque, and that's kind of the brain's defense against further onslaughts from inflammation or heavy metals or other stressors.
Yes and no, but I don't want to go into the details. Dale's a friend of mine. We have our names on a few papers together. I was at University of Kentucky for 11 years at the Center on Aging, and Dale's from Kentucky. But I'm very much on board with his general idea, and we need to reduce risk, and we have some good ideas. The nutrition standpoint, I kind of have not exactly the same angle on it.
What's your take on nutrition and Alzheimer's disease? Let's get that in. So here's some really good ideas around food and Alzheimer's prevention.
Yeah, so here's my own thinking on this. I think variety in diet is good. Vegetables, a lot of vegetables, some fruits, nuts are good. Fish, not red meat. I'm not real keen on ketogenic diets that have a lot of red meat, and then olive oil instead of lard, whatever, so we did a lot of work on this. It's very interesting, the beginning with Linus Pauling, and then down the road, this notion came out, you can load up on antioxidants and, you're not going to get cancer, you're not going to get heart disease. And
so for those of you at home, Linus Pauling discovered vitamin C, ascorbic acid. He was known for taking approximately 6 grams a day, which is a huge dose, and he lived to be 94 years old, also won the Nobel Prize.
Yeah. But he kept his mind very active, and he walked every day. And I think he ate a pretty healthy diet. But anyway, so my view on what's good in vegetables and fruits is actually chemicals that evolved as toxins or chemicals to keep insects and herbivores, omnivores from eating the plants. So things like sulforaphane and these good chemicals act through hormesis-based mechanisms. They actually impose stress on our cells.
So we're not talking polyphenols.
No, we're not talking about chemicals that directly scavenge free radicals. We're talking about chemicals that stimulate our cells to produce their own intrinsic antioxidant defenses.
So these slightly stress our body when we eat them, and that has a beneficial effect. Now, are these the same as what Dr. Stephen Gundry refers to as lectins that are found in plants that can cause mild to moderate inflammation?
No, there's a variety of different chemicals. There's terpenoids, alkaloids, polyphenols, a number of them. I wrote an article for Scientific American in 2015 July issue, and the whole point of the article was that the chemicals in fruits and vegetables that are good for our health are actually toxins from an evolutionary perspective. They're concentrated in the skin of fruits, the delicate parts of vegetables, sprouts. The most early, if the insect or eats a sprout, that's it. The whole plant's dead, right? So these chemicals that are concentrated in the vulnerable parts of the plants, their function is to keep insects and other organisms from eating the plants. And it turns out, as the plant grows and the fruit, say we take fruits, for example, as the fruits ripen, the concentration of those chemicals decreases. We did some work on a chemical called, well, people have heard of sulforaphane, curcumin, which is in turmeric root, and caffeine. Caffeine is actually, if you put caffeine on your tongue, pure caffeine, it's very bitter taste. All these chemicals have very bitter taste. The pure chemical, you wouldn't want to eat very much of yourself. But tomatoes, so they start out as green tomatoes, right? And then they turn red. So it turns out there's this chemical tomatidine that's high concentration in the green tomatoes. And as it ripens, the concentration decreases a lot. And this chemical, very bitter taste, it keeps insects from eating the green tomatoes, or birds from eating the green tomatoes. Then when they get ripe, from an evolutionary perspective, it's not only not bad, it might be a good thing if a bird eats the tomato and the seeds, because then it'll fly over there, poop the seed out, and you'll get, these plants are very smart. Yeah. And distribute. But this coevolution, so the point is there's this coevolution between plants and animals that I think helps explain why vegetables and fruits are good. So a lot of fruits, not a lot of fruit, a lot of vegetables, some fruits, and particularly kind of the parts of the fruits that other people don't like to eat because they're bitter tasting.
Like, for example?
Skins.
Great.
So I have, like my wife makes apple pie. She skins the apples and I eat all the skins.
There you go. Great fiber there too. Thank you. That's, these are such important practical steps of behaviors and foods to eat. I don't know if Hippocrates said this, but he's credited as saying, let food be thy medicine and medicine be thy food. Hippocrates, the grandfather or father of modern medicine. I want to ask you a quick clarifying question. Do you think the research shows that does GABA cross the blood-brain barrier? I haven't gotten a clear answer from the research.
Yeah, I think it can cross the blood-brain barrier. It's a sense. So this is another interesting thing. GABA is produced from glutamate. So glutamate's an amino acid. It's a building block of proteins like the other 19 amino acids. That's what most people know amino acids for. Most of the neurotransmitters that we know about are derived from amino acids. Glutamate itself is an amino acid. GABA is just a slight chemical modification that cells make. There's a enzyme called glutamate decarboxylase that removes some atoms from the glutamate molecule, and that makes GABA. So that's in GABAergic neurons.
So GABA is probably small enough to cross the blood-brain.
Yeah, it definitely is. And the other transmitters too. So dopamine, serotonin, norepinephrine, they're produced from amino acids, right? Serotonin's produced from the amino acid tryptophan. Norepinephrine and dopamine are produced from the amino acid tyrosine. So this is another, in my book, Sculptor and Destroyer, I talk about the evolution of glutamate as a neurotransmitter and how it was probably the first neurotransmitter. If you go back,
Ooh,
it has roles even in bacteria,
wow.
in communication between bacteria.
I would like to take a brief break and acknowledge our sponsor, Air Doctor. So it is a really good idea to get an air filter for your home, your office, and especially your bedroom, because our air is not clean, and an unclean air can bring toxins into our body, and that can hurt our brain. Dr. Dale Bredesen talks about the importance of getting a HEPA filter. Now, Air Doctor is a very sophisticated air filter. It filters out 10 particles 100 times smaller than a regular HEPA filter. So it's very high quality. If you want to try an Air Doctor, it is a great product. I use it. I have one right next to me. I have one in my bedroom when I sleep, and I use it next to me in my office when I work during the day. So if you'd like to try an Air Doctor, go to airdoctorpro.com/love. That will give you a really nice discount.
I had two questions I want to ask. One question is, I'd love to hear the difference between your understanding of amyloid versus tau. So amyloid beta is the plaque associated with Alzheimer's disease. Then we hear about tau tangles. I wonder if you could explain, my understanding is amyloid is between cells, tau tangles is inside the cells. Any other important differences for people to understand, because these are often lumped together. I don't know if that's helpful for people's understanding of Alzheimer's disease. Could you just talk about the difference between tau and amyloid briefly?
Well, their accumulation outside of neurons with amyloid and inside of neurons with tau, you don't see that in brains of young people. So it's an age-related thing. The amyloid beta peptide, the protein that's commonly referred to as amyloid and amyloid plaques, comes from a much larger protein. Scientists tend to use logical names oftentimes. So the amyloid precursor protein is the protein from which the amyloid that accumulates outside cells in Alzheimer's disease comes from. This amyloid precursor protein has very important functions. Actually, we had evidence. It's very interesting. It quiets neurons in a manner kind of similar to GABA. We think its normal function is to kind of help kind of subtly modify synaptic activity. Okay, so the tau protein is a protein inside of neurons. Its normal function is to control what's called the molecular skeleton of the neurons. There's these polymers of proteins that form what are called microtubules that are distributed within the neuron. So they give the neuron its form. So here's a neuron. So I guess the people have to go to your YouTube channel.
That's a big neuron.
It's a big neuron. So anyway, this is an axon here, the long process. These are dendrites. These receive information. These send signal to the next neuron. So within these axon, these microtubules give it the structure that keep it like that. Otherwise, it would be, like that.
So tau provides structure or architecture inside the neuron. Is it particular to the axon, or is it throughout the whole neuron?
This is a good question. It's normally only in the axon, but in Alzheimer's disease, it kind of falls out of the axon and goes into the cell body and even the dendrites of the neuron.
So the axon's the long extension.
Yeah.
And so that long extension has architecture inside of it. That architecture is tau. And in Alzheimer's disease, that architecture can get messed up, go inside the cell body or into the dendrites. And then does the axon then not function? Does the the neuron doesn't function as well then because it's missing that architecture, and the axon might go to, stimulate different neurons because it doesn't, or it might not function at all.
Yeah.
Is that right? Does the axon no longer fire correctly because it's missing the architecture, the support?
Yeah, essentially, there's changes going on both the axon and the dendrites, because remember this excitotoxicity component too. A neuron where this tau starts to fall off the microtubules and get in the cell body. At the same time, the neurons are being stimulated by glutamate in the dendrites. And so you kind of get simultaneous bad things going on in both the axon and the dendrites. And the tau, by the time neurons have accumulated a lot of tau, they're pretty much spent. They can't, they're already severely compromised.
So they're not firing correctly. They're not functioning properly. What's the solution then? Is the solution, apoptosis and then neurogenesis? Apoptosis, listener, is the planned cell death is the cleansing of old cells and then potentially making room for new cells.
In an ideal world, that would be an answer. But unfortunately, I mentioned there's only 2 brain regions where there's stem cells that can form new neurons. And so most of our brain, the neurons, if they die, they can't be replaced. It's only in the hippocampus that some neurons can be replaced. And there it's only a subset of the neurons that can be replaced.
So once a neuron is infected or affected by dysfunctional tau, the tau tangles, it's no longer functioning well and it needs to be removed. Otherwise,
it's too late.
It's probably creating inflammation as well. And
absolutely. But when neurons die, you get inflammation.
Okay, very interesting. And so that's what happens with tau. And so tau accumulates not just in the neurotransmitters in the hippocampus, but it sounds like throughout the brain.
Yes.
And then what's your thoughts on amyloid? What do people need to know about amyloid to understand its role? Because the research has been dominated by amyloid since the 80s, I think.
So my view on this is that changes that occur during normal aging, things that have the potential to lead to amyloid accumulation, tau abnormalities, Alzheimer's disease. All of us are at risk for Alzheimer's disease. Now, depending on our lifestyle, and diet, stress level, bad stress levels, and so on, we can counteract age-related problems that can lead to downstream accumulation of amyloid, the formation of the tau neurofibrillary tangles. Some of these changes that occur during normal aging, I mentioned the mitochondria tend not to function as well. And there also tends to be accumulation of molecules damaged by free radicals. Proteins, DNA, and membrane components, the lipids in the membrane of cells, can all be damaged by free radicals. And normally cells have a way of repairing the damaged DNA, getting rid of damaged proteins, getting rid of damaged membrane lipids. But during normal aging, in everybody, the ability of cells to repair or remove the damage is compromised. That ability to remove or repair the damage can be further compromised by overeating, being sedentary, not keeping your brain intellectually challenged or socially engaged, and maybe some specific dietary factors that we've talked a little bit about. And then, on the other hand, healthy lifestyles can counteract those. So there's better DNA repair. There's better ability to clear out damaged proteins and lipids through a process called autophagy. Yeah, and then again, the switching back and forth between the challenge of these healthy lifestyle factors and recovery periods is very important too. So that switching back and forth throughout life. stress recovery.
So the unhealthy lifestyles promote the accumulation of amyloid.
Yeah. So that if you don't subject your cells to stress, their ability to deal with stress is worsened, it's compromised. So again, if you're a cell and the individual you're living in is, eating breakfast, lunch, dinner, snack every day, overeating, if they're not getting any exercise, in the case of neurons, if they're, not doing much serious thinking every day, watching the boob tube, the cells are not challenged. And so their ability to get rid of the molecular damage to the proteins, DNA, and lipid or repair it's compromised. And so aging, it's just like a perfect storm. Aging in someone who has poor diet and lifestyle, unhealthy diet and lifestyle.
Very interesting. You were head of neuroscience for the National Institute on Aging. I would love to hear your favorite anti-aging longevity strategies based upon the research. So what do people need to know about slowing down aging?
So you mean besides what we've already talked about?
So so let's review that. So we've talked about exercise, and then we've talked about diet, and then healthy lifestyle. We didn't mention sleep, but sleep is very important as well, and I understand that's really key in the literature. And any other thoughts on slowing down aging?
Yeah, there has been, when we've been involved in some of this, a question comes up: can you mimic these beneficial effects of exercise, intermittent fasting, intellectual challenges pharmacologically? Is there some chemical that will mimic the effect? And the answer is, there are some chemicals that will partially activate these adaptive stress response pathways, or in some cases, quite a bit of overlap. So we had this idea that, okay, let's subject animals intermittently to some chemical that we know imposes a stress on the cell. So one thing we used is called 2-deoxyglucose. It's essentially a modified form of glucose that gets into cells but cannot be used to produce ATP. So it's essentially the equivalent of doing fasting. If you give this to animals, it's essentially like they go into this fasting-like state, at least somewhat. It's not exactly, but from the standpoint of ketones, for example, the ketones will go up because the system's tricked into thinking that glucose levels are low. And so ketone levels do go up. We found some neuroprotective effect. Another one was a drug that imposes a stress on mitochondria. It's called a mitochondrial uncoupler. DNP is the name of the chemical. And I should say, for all these things I'm telling you, it turns out there's a limited level of doses where we see any beneficial effect. Lower doses, we don't see a beneficial effect. Higher doses, we see detrimental effects. So high doses of these chemicals are bad.
Now is metformin a mitochondrial uncoupler?
No, it acts in a different manner.
But it does stress the mitochondria, is my understanding.
Yeah, not as much as DNP. It's much more mild.
Okay.
And then another one you and your listeners may have heard about is rapamycin. And that inhibits a pathway called the mTOR pathway. Fasting is like a really potent inhibitor of the mTOR pathway. The NIA, beginning quite a while ago, the National Institute on Aging funded these very large multi-center studies, 3 different centers in the United States did the exact same experiments and looked at, can we identify chemicals that increase lifespan in rats or mice? Okay. And most of the chemicals they tested didn't, including, actually, we were involved in that, 2DG, 2-deoxyglucose didn't. It turned out it had some bad effects on the heart. But rapamycin was one where there was a significant extension of the average lifespan in the mice at all three centers. The effect was not dramatic. It wasn't like with calorie restriction or intermittent fasting where we see a doubling of the lifespan. This is more like 10%.
Now, has that been replicated in humans, or is that just in animal studies?
No, so that's the whole thing with aging research. Lifespan studies this kind of study is pretty much impractical in humans because you have to start it when someone, say, ideally when they're young, but certainly by mid-age, and then keep treating them over decades and decades and decades until they start dying, right? The end point is death. So there's a study with dogs going up at University of Washington on lifespan effects of rapamycin.
Oh, interesting. Now,
it turns out there's a big...
Go ahead, please.
Yeah, the pet story is very interesting too. So now people have pets that have short lifespans because they overeat and don't get any exercise. And so pet owners would like to be able to put something in the food of their dog and have it be healthy. But just feed it once a day, and, which is normal for wolves, right? They evolved from wolves. Give it plenty of water, feed it once a day, and let it get some exercise, and that's going to be the healthiest for your dog.
So longevity for animals, for your pets, feed them once a day and plenty of water, and give them the opportunity to exercise.
Yeah,
that's terrific. What are your thoughts on what's your interpretation of the data on SIRT1 agonists, things that increase silent information regulator 1, like NMN, NR, and then there's even data on quercetin and resveratrol, that those can potentially have anti-aging effects, as well as the anti-inflammatories like curcumin may have anti-aging effects because, generally speaking, inflammation is a risk factor for aging, for the brain as well as for the skin. What are your thoughts on both SIRT1 activators and anti-inflammatories for anti-aging?
There's a lot of data on these in animals, but not so much on lifespan, except in worms and yeast. So David Sinclair started out in yeast, right? These single cell organisms, looking at lifespan, and in those animals, the sirtuin activators. Well, I don't know. I have a little pet peeve with the way they actually measure lifespan. It's just they measure lifespan by population doublings. How long can the cell keep dividing? But in worms, C. elegans, there are some data suggesting they can increase lifespan, and mechanisms of action. There's quite a bit of data that would give a rationale that these do good things to cells that would be consistent with an anti-aging effect. There's been work on resveratrol at the NIA.
That's been rather controversial. People talk about it hasn't been replicated in humans, or the studies were tainted. What are your thoughts on resveratrol?
I guess I'd say, for resveratrol, it's not going to hurt you for sure. I don't know of any data that has, it's a natural product, red grapes and so on. And so I think it's fine. It does activate some adaptive stress response pathways. So in theory, it should have some beneficial effects. Quercetin, now you're getting into and we did a little work on this on cell senescence, right? The senolytics. As we age, there's these cells that stop dividing, and then they get big and they start causing a lot of inflammation. And these senescent cells, there's a couple things that have been shown to remove them in animal studies and in humans, actually, in skin cells. One is quercetin, and another is dasatinib, or it's actually a drug. You can't get it. It's a prescription drug.
Now, fasting also helps remove senescent cells,
Yeah.
doesn't it? So,
Yes,
so
it does.
quercetin has a similar effect as fasting
Yeah.
as far as removing. Is it autophagy? Is quercetin increasing autophagy, and that's what's getting rid of senescent cells, clearing of senescent cells?
Yeah, that's part of it. And it may, it's still kind of unclear exactly how the senescent cells are removed, but probably they undergo apoptosis. It probably triggers apoptosis, and then they're removed by macrophages.
Very interesting. So quercetin shows the ability to help remove senescent cells. Resveratrol is unlikely to be harmful and may be a slight stressor, may have a hormetic effect, so that can be beneficial. And what do you think of NMN and NR? NMN is now very difficult to find in the United States. It's still available in Canada. NR is readily available. What are your thoughts on these anti-aging?
We were involved in some of these studies in a collaboration in a mouse model of Alzheimer's disease that we developed. There's a colleague at NIA. His name is Will Bohr, Bohr.
And any relationship to Niels Bohr?
The grandson.
Really? Oh, that's so cool. So the Bohr model of the atom.
Yeah,
that's the grandson.
And his father, Will Bohr's father, so Niels Bohr's son, also got a Nobel Prize.
Wow. I wonder how many father-son or mother-daughter duos got Nobel Prizes.
That's the only one I know, but there may be more.
Wow. And then side note, trivia, Marie Curie, I think, is one of the only people to have, she had 2 Nobel Prizes, one in chemistry and one I think in biology, or it might have been in physics, but she was not inducted into the National Institute of Science in Europe. So she had 2 Nobel Prizes, but she couldn't get it into the club elected by her colleagues.
Well, it's interesting you use the word club, because it is the National Academy of Science and the equivalent in Europe. It's a little bit of a club.
Yeah, a bunch of men that were, probably didn't like being outperformed by a woman.
Yeah. And but anyway,
so
the bottom line is we give these Alzheimer's mice that have amyloid, tau pathology, and some neural loss. We give them NR, nicotinamide riboside, long term for months and months and months, and it does lessen the pathology and lessen the cognitive deficits. We published that paper a number of years ago, and there's other labs.
What the dosage was? What a dosage for a healthy adult might be? So like 1,000 to 2,000 milligrams?
Well, no. So we got to go by body weight, right? We're giving it to mice.
Okay.
So I don't remember off the top of my head. I can send you.
I'd love to know that. And the ability to translate mouse data into humans, I think is important, because we don't want to take the same dosage as mice, because those dosages are massive, would be just massive for humans, milligrams.
No. But in general, if you milligrams per kilogram body weight, then that's kind of the way you want to think about it. So if we give a mouse 1 milligram, a mouse weighs 30 grams. So that would be, I don't know the math, 1/50th of the, even less, 1/100, whatever, of a human dose. So we'd actually, if we give, anyway, in animal studies, we always adjust it to the body weight. It's interesting, humans, that isn't often done. I think it actually should be done.
Yeah, that make a lot of sense. But it's not a 1-to-1 conversion, right? So 1 milligram per kilogram in mice does not mean a human should be taking 1 milligram per kilogram, for example.
No, but that would be a good starting point.
Okay. So NR, whatever dosage it would be in humans, you found that NR, or the data shows that NR helps, is it slow cognitive decline or helps reverse cognitive decline?
No, slow cognitive decline.
Okay, that's really interesting, and that's not a pharmaceutical This is something that I don't want to say it upsets me. This is something that intrigues me that I think this is also on your radar. There's so many supplements and behaviors that are very helpful in preventing Alzheimer's disease. And yet all the news is about, this new drug slows it by 25%. Well, how much does NR slow it, right? Potentially more, and it's a lot safer, and it's available for everyone. You can order it right now, and it doesn't cause brain bleeding or doesn't have the risk factor of brain bleeding.
What happens, Robert, is the following. It takes a lot of money to run a clinical trial in humans, right? It's mostly paying staff, right? The people that perform the trials, or you could do MRI or cognitive testing and so on. And so if there's something like nicotinamide or NR or resveratrol or something that people that can buy over the counter, it's really comes to the NIH. They're the only ones that has the money to actually do the funding for that. Pharmaceutical companies aren't going to put in any money because they actually don't want those studies.
Right, and they don't have a patent for it. It's not part of their business model.
No.
But how do we make this part of the health model in the United States, right? Who's going to fund research on non-patentable compounds that have the potential to slow down aging and prevent Alzheimer's disease?
Yeah, unfortunately, the NIH does some. And for example, intermittent fasting, I was very gratified, because I was at NIA for 20 years and doing intermittent fasting work all that time. And so I knew all the administration at NIA and the NIH and Francis Collins and so on. And so it was very gratifying. They knew my work was good and they were confident in it. And so for the last 6 or 7 years, they've been putting a lot of money into clinical trials of intermittent fasting in humans. If you go on clinicaltrials.gov, if anyone wants to know what clinical trials are going on anywhere, clinicaltrials.gov, you can put in resveratrol, Alzheimer's, or whatever, and find them. But right now there's over 150 clinical trials of intermittent fasting, cancer patients, type 2 diabetes,
Great.
a lot of inflammatory disorders, some neurological disorders, recovery from stroke, a lot of different things. And that's the only funding source for human clinical trials of intermittent fasting is NIH. Because there's no company. How do you make, and that's the problem. The things that are good for your health, people can't make money so easily on. So you've got this big healthcare industry, right? It's built around physicians and this historical scenario where you work, pharmaceutical companies develop, government-funded scientists develop drugs, show that they have an effect in animals. Then a drug company licenses that molecule, gets actually patent protection, and then they fund clinical trials, but that can also be bad, right? Because they have a bias, right? So, yeah, the system is not ideal, and it doesn't give the public a lot of confidence in the system when I think there's got to be some, I'm not I guess what you'd call a pure socialist, but it would be better if this wasn't all, our healthcare wasn't all profit driven, and these countries, Sweden and Denmark, those countries where the whole healthcare system is through taxpayers' money, it seems to work pretty good.
Yeah, I mean, I would love to see clinical trials, double-blind, placebo-controlled clinical trials in humans of curcumin, of NMN, of Ceylon cinnamon, just to name a couple of herbs, of lion's mane, and then just have them take cognitive tests for 6 months. This can be done online. The supplements are safe. They're relatively inexpensive. And I understand there's not a lot of profit at the end of that trial, but then we'd have some data to show that, listen, in healthy adults and those with early stage Alzheimer's disease, here's the benefit of these natural herbs. And then let's educate people about this. That way they can make informed, healthy decisions.
So you know what will happen? So this is going to happen with psilocybin. So, mushrooms lie in Maine. There's some chemicals in there that are good for health, and scientists will identify like these specific chemicals are what's giving the lion's mane this effect. Just like psilocybin, yeah, it's the same thing that gives you hallucinations that's effective in patients with depression, or it turns out it looks like addiction is. People with addiction are also going to benefit.
Oh my goodness. I love the research out of Johns Hopkins showing one session of psilocybin mushrooms, a psychedelic dose of psilocybin mushrooms with a psychedelic psychotherapist stopped smoking 80% of the time. So 8 out of 10 people, it helps them stop smoking, which is amazing, because can you imagine a stronger addiction to break with one session? And then my guess is this will probably be true with alcohol. It might be true with sugar addiction. Who knows?
Maybe even obesity.
Oh, goodness gracious. Wouldn't that be wonderful?
Food addiction.
I imagine Big Pharma is very scared.
Here's what's going to happen. Here's my prediction.
Yeah.
Okay, so we can just grow the mushrooms and take the mushrooms, right? Right? We don't actually. These things are, you can standardize this. So, if you take one mushroom or two mushrooms, that's equivalent to this exact dose of psilocybin. It's kind of like what's done with marijuana, right? So there are companies, I know of companies that they are taking the psilocybin molecule and they're tweaking it a little bit. And maybe even, changing it in a way that doesn't change its effect at all, but it's a different molecule. And then they're going to do a clinical trial in depression and say, look at our molecule. And then it's not, when it comes out on market, it's not going to be a name anybody recognizes. They'll make up some name so that the general public doesn't know, oh, this is just essentially psilocybin because the general public, unfortunately, this country is kind of science illiterate overall. And so kind of general public doesn't know a lot of what's going on, these kind of details. So like these Alzheimer's antibodies, people, oh, this is a new drug for all. No, it's just there's another amyloid antibody.
So what Big Pharma is going to do is they're going to compete with nature. So there's clinical trials out of Johns Hopkins, out of Stanford. By the way, I love that mainstream neuroscience is taking on psychedelics as research. I think it's really wonderful. And 10 years ago, 5 years ago, people could lose their jobs for talking about it, and now people are getting grants for it, which is just wonderful. Yeah. And so there's data to show that psychedelics are, especially psilocybin, are very, very helpful for ending addiction. And your prediction, and it sounds like you have a little insider information, is that pharmaceutical companies are taking the psilocybin molecule, tweaking it just a little bit, and they're going to run clinical trials saying, look, we got an antidepressant drug, which could be very helpful. So that theoretically could be a wonderful thing. But what they're competing with nature, what Mother Earth made, or what, however it was made. And now they're going to compete with that. And they may try, I'm guessing, they may lobby to keep psilocybin illegal in some states, because if that's legal, and it probably has easier access to it than a pharmaceutical drug. It might be less expensive and so forth.
I think it's going to, so it's legal in Oregon, or decriminalized in Oregon. It's decriminalized in Washington, D.C. There's
D.C. or Washington State.
Colorado, there's going to, I think in the next election, they're going to have a bill or
so. I was slightly active in the campaign in Colorado. A friend of mine was very active. His name's Rumsey. He helped get Colorado to decriminalize plant-based entheogens. So they're decriminalized. So it's legal to possess. It's legal to it's legal for psychedelic psychotherapists to use psilocybin and other plant-based entheogens in their practice in Colorado now. And it's really exciting. I want to thank Colorado. They're also the first state to legalize recreational cannabis, which is a big step. So Colorado really on the cutting edge.
I think for psychedelics, that's a reasonable approach to it, though, is, it's legal because it turns out it is important to be kind of in the right environment and, when you're taking these things. So I think as long as they aren't charging, when they start actually charging patients, they don't charge them very much.
Right. So therapists are allowed to charge what would be a fair-based exchange, because a therapist has to acquire the mushrooms. And so if they're using psilocybin or ketamine in their practice, there has to be some fair exchange for that. So the law does allow for you to set up a shop, but the law does allow for therapists to use it in their therapy, and then charge clients a reasonable fee to use that in their therapy, which is really wonderful. Dr. Mattson, this has been so fun. We scheduled an hour. We're almost at 2 here.